Hi viewers, welcome to SEKHAR'S Biofactory for NEAT. Sexual reproduction in flowering plants with anatomy of anther. If you like this video, subscribe my channel, like, follow and share my videos to your students and friends to get more information on NEAT and other competitive exams. In this video, I am going to explain the morphology and anatomy of male reproductive part in a flower. Morphologically, every stamen technically is called Microsporophyll. In a flower, the group of stamens are called androecium and each stamen is considered as male reproductive part. In the structure of every stamen, two parts are present. One is called filament and other one is called anther. The filament is a long slender stalk of stamen and it is sterile in nature because it never produces any pollen grains. This filament generally it having two terminal ends. One is called proximal end and other end is called distal end. The proximal end of filament it is attached to thalamus. Sometimes it attaches to petals in a flower. But the distal end of a filament it attaches to anther. Anther Anther is the main fertile part in stamen and it is the site for pollen production. In most of the angiosperms, the anther having two lobes in its structure. This two-lobed anther is called bilobed or dithecous anther. In each dithecous anther, four microsporangiums are present in its structure so that it is also called quadrilacular anther. The dithecous anthers are observed very clearly at earlier stages only before maturation, but very rarely in few plants which belongs to Malvaceae family. The anthers having only one single lobe. Such anthers are called unilocular or monothecous anther. But very rarely in few plants like Acacia and Canna. In these plants, the filament it having one unfertile or sterile anther. Now such type of stamens are called staminodes. In these staminodes, anther never produce any pollen grain. In the structure of each stamen, filament and anther are present. In this anther, every lobe is called theca. This lobe contains pollen sacs, especially each lobe having two pollen sacs. In dithecous anther, totally four pollen sacs or microsporangiums are present. In monothecous anther, two pollen sacs or microsporangiums are present in its structure. To understand the internal structure of anther, we have to take the transverse section of anther. In transverse section of young dithecous anther, it having two lobes or these lobes are called theca. These two lobes are connected with a tissue. It is called connective tissue. It is equal to vascular strand. In each lobe, two pollen sacs or microsporangiums are present. These four microsporangiums are present at corners. In the internal structure of anther, each microsporangium is surrounded by anther wall with four layers. The first layer is called epidermis, endothecium, middle layers and tapetum. In the structure of young Dithecus anther, the total anther is surrounded by a single layer of epidermis and it is outermost layer. Below to this epidermis, a single layer of epidermis endothelium is present. Below its endothelium, middle layers are present around the microsporangium. The last layer in anther wall is tapetum. This tapetum present around microsporangium. In each microsporangium, after maturation, a number of pollen grains are produced. Now, when we observe the microsporangium structure with anther wall, the microsporangium contains anther wall with 4 layers. The outermost layer is epidermis, second layer is endothelium, third one is middle layers, and the last layer is tapetum. Inside microsporangium, many pollen grains are generally developed. Let us discuss each and every anther wall layer in detail. Epidermis is the outermost layer present in anther wall with single layer of cells. This epidermis is present around the anther. The main function of this epidermis is protective function. The second layer is endothelium, a single layer of radially elongated cells. These are present just below to the epidermis. It is called endothelium. In this endothelium, each cell it having fibrous thickenings in tangential cell walls. Now these fibrous thickenings are made up of a special chemical. It is called alpha cellulose. The main function of this endothelium is protection and dehiscence of anther, because the endothelium cells are hygroscopic in nature. During maturation, this endothelium it helps to break the entire anther wall to release pollen grains. The most important character, the portion which present in between two microsporangiums in a theca. In this portion, the endothelium cells lack fibrous thickenings. Now such locations are called stomium. The third layer is middle layers. Below to the endothelium, 1 to 3 layers of middle layer cells are present around microsporangium. The main function of these middle layers is for protection and also it helpful for dehiscence of anther. The first 3 layers, epidermis, endothelium and middle layers are initially it protects the anther and also it helps in dehiscence or breakdown of anther wall after pollen maturation. The last layer in anther wall is tapetum. It is the innermost layer of anther wall. It present around microsporangium. In this tapetum, only a single layer of elongated cells are present. The main characters of this tapetum is each cell in tapetum it is radially elongated, and in the structure of every cell a dense cytoplasm is present with nutrients. In every cell more than one nucleus is present. That condition is called multinucleate condition. In each tapetum cell, the multinucleate condition is developed due to endomitosis process. In this endomitosis process, the nucleus undergoes division inside the nuclear membrane. At that time, cytokinesis is completely absent. Due to mitosis without cytokinesis, it leads to formation of more than one nucleus in a cell. That leads to multinucleate condition. At the same time, each nucleus, it is polyploidy due to endoploidy nature. The main characters of tapetum: it performs endomitosis. Normal mitosis takes place not followed by cytokinesis, formation of a restitution of nuclei, endo reduplication of chromatin material. The tapetum, it is basically two types. One is called Amoeboid tapetum, Secretory tapetum. In the structure of Amoeboid tapetum, this Amoeboid tapetum is also called invasive or plasmodial type. In this type, the tapetum cell having a cell wall, this cell wall, it will be dissolved. After dissolving the cell wall around the tapetum cells, it contains protoplast with cell membrane only. This tapetum cell, to provide food material to developing pollen grains, they fuse and move in between the pollen grains. Now at this time it appeared with amoeboidal structure, so that it is called amoeboidal tapetum. In amoeboidal tapetum, it is the most effective way to supply food materials to developing pollen grains directly. It is the microscopic view of amoeboid tapetum. The second type is secretory tapetum. In this secretory tapetum, it is also called granular or secretory tapetum. It is the most common type of tapetum seen in most of the angiosperms. In this type of tapetum, the tapetum cells are remain present in their original position, but due to breaking of their cells progressively, the substances are released to the developing pollen grains. Now these secretory tapetum having various functions. It is the microscopic view of secretory tapetum. Coming to functions of tapetum, the most important function is nourishment. The tapetum, it supply food materials to the developing microspore mother cells and pollen grains. The tapetum, it helps in the formation of pollen wall around pollen grains. At this time, some of the structures called eupsic bodies. These eupsic bodies are a small acellular structures of sporopollenin. These materials are present in cytoplasm of tapetum cells. During pollen grain wall formation, when the tapetum is break down, these acellular sporopollenin structures get deposited around pollen grain that leads to the formation of pollen wall. These structures contain carotenoids. These carotenoids can form pollen kit around pollen grains. The pollen kit is nothing but it is a yellow sticky gum-like substance with oily nature. It is deposited around the pollen grain. Now it is called pollen kit. The main function of this pollen kit is, due to its sticky nature, especially in insect pollination, the pollen grains can easily attach to body of insects. Along with that, when pollen grains are deposited on stigma, because of this pollen kit and sticky nature, it can easily hold on stigmatic surface. Along with that, the most important function of tapetum is tapetum cell can secrete calase enzyme. The chemical nature of calase enzyme is beta-1,3 gluconase. This calase enzyme, generally, it helps to disintegration of calase present in between pollen grains. Initially, after completion of pollen development, the pollen tetrads are collectively present because of calase, one of the material. This calase enzyme it can break down the material present in between pollen grains in a pollen tetrad. Then, after disintegration of this material, pollen grains can easily separate it to each other. That is the function of Calais enzyme. It is developed from tapetal cell. During microsporogenesis, tapetum cells can produce various enzymes, hormones, and along with amino acids and other nutritional materials. All these are required for the development of pollen grains. Along with that, auxin, it is produced from sporophytic microsporophyte cells. Other than tapetum cells, it is required at earlier stage for the development of pollen grains. Now coming to structure of Microsporangium or Pollensac. In Dithecus anther, totally 4 microsporangiums or pollen sacs are present at 4 corners. In each microsporangium, it is filled with homogeneous sporogenous tissue. This tissue is diploid in nature. Each sporogenous cell, it becomes into microspore mother cell. This microspore mother cell, it have capability to produce 4 pollen grains or microspores with the help of meiotic division. Every sporogenous cell, it becomes into microspore mother cell, then it can produce 4 microspores or pollen grains. After completion of meiosis division, the pollen tetrads are formed in Microsporangium by utilizing the food substances from tapetum cells, and tapetum completely degenerates. After completion of pollen production in every Microsporangium, the anther, it undergo maturation. The tissues present in between 2 microsporangiums of a theca or lobe, it will degenerate, and 2 microsporangiums will be fused to form a chamber. The pollen grains are partially stored in these chambers before releasing. After completion of maturation, the anther wall, it starts its dehiscence at stromium. During this dehiscence process, epidermis, endothelium, and middle layer cells are participated for dehiscence or breakdown of anther wall. After breakdown of anther wall, the pollen grains are released to outside. Now it is the structure, internal structure and maturation of anther. Thank you for watching. If you like this video, subscribe my channel, like and share my videos to your students and friends.